cell cycle is a fascinating process that governs how cells grow, replicate, and divide. It's essential for everything from development to tissue repair. Let's break it down into its key phases: interphase and the mitotic phase. Interphase is where the magic begins. It's the longest phase of the cell cycle, accounting for about 90% of the time a cell spends in the cycle. Interphase itself is divided into three stages: G1, S, and G2. During G1, or the first gap phase, the cell is busy growing and performing its normal functions. It's like a student preparing for an exam, gathering all the necessary materials and information. The cell increases in size, synthesizes proteins, and produces organelles. Next comes the S phase, or synthesis phase. This is where the real action happens. The cell duplicates its DNA, ensuring that each daughter cell will have an identical set of chromosomes. Think of it as making a photocopy of a textbook. Every page needs to be copied accurately. After the S phase, we enter G2, the second gap phase. Here, the cell continues to grow and prepares for division. It checks for any DNA damage and makes repairs if necessary. This is crucial because any errors in DNA replication can lead to serious problems, including cancer. Once interphase is complete, the cell is ready for the mitotic phase, which consists of mitosis and cytokinesis. Mitosis is the process where the cell's nucleus divides, and it's broken down into several stages: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses into visible chromosomes, and the nuclear envelope begins to break down. It's like getting ready for a big performance. Everything needs to be in place. During metaphase, the chromosomes line up in the middle of the cell, attached to spindle fibers. This alignment ensures that when the chromosomes are pulled apart, each new cell will receive an identical set. Anaphase follows, where the sister chromatids are pulled apart to opposite ends of the cell. This is a critical moment. If something goes wrong here, it can lead to cells with an abnormal number of chromosomes. Finally, we reach telophase, where the chromosomes begin to decondense back into chromatin, and the nuclear envelope reforms around each set of chromosomes. It's like the curtain coming down after a show, signaling the end of one act and the beginning of another. Cytokinesis then occurs, which is the physical division of the cytoplasm, resulting in two separate daughter cells. In animal cells, this happens through a process called cleavage furrow formation, while in plant cells, a cell plate forms to create a new cell wall. Understanding the cell cycle is crucial because it lays the foundation for how organisms grow, develop, and maintain their tissues. Disruptions in this cycle can lead to diseases, including cancer, making it a vital area of study in biology and medicine. So the next time you think about cells, remember the intricate dance of the cell cycle, where each phase plays a pivotal role in the life of a cell. It's a remarkable process that keeps life moving forward, one cell
What
at a
is the
time.
cell cycle? It's the journey a cell takes as it grows, prepares, and divides into two new cells. Now, karyokinesis? That's the fancy term for the nucleus splitting during cell division. Variations are the unique differences we see among individuals of the same species. Ever heard of crossing over? It's when non-sister chromatids swap genetic material during meiosis, adding to genetic diversity. A nucleosome. Think of it as DNA's packaging unit, with DNA wrapped around histone proteins. A gene is simply a segment of DNA that holds the instructions for making a specific protein. And finally, a nucleotide is the fundamental building block of DNA, made up of a sugar, phosphate group, and a nitrogenous base. Mitosis is all about making identical cells, while meiosis creates genetic diversity with half the chromosome number. Think of it as a cellular shuffle. Now, haploid cells have one set of chromosomes, like a single player in a game, while diploid cells have two sets, ready for a team effort. In metaphase, chromosomes line up in the center, but in anaphase, they split and move to opposite sides. It's like a race. G1 phase is all about growth and preparation, while G2 phase is the final check before cell division. Cytokinesis is the actual splitting of the cell, whereas karyokinesis is the division of the nucleus. Chiasma forms during crossing over, where chromosomes exchange genetic material. It's nature's way of mixing things up. Chromosomes are the X-shaped structures carrying genes, while chromatids are the individual strands before they split. Finally, the centromere is the region where chromatids are joined, and the centrosome is the hub that organizes microtubules for cell division. Each term plays a vital role in the dance of
Did
life.
you know that a mouse has 40 chromosomes in its somatic cells? That means it gets 20 from its father. When it comes to gametes, they only have 19 autosomes, plus one sex chromosome in the ovum. Speaking of chromosomes, during mitosis they line up in metaphase, and by telophase, the nuclear membrane reappears. Prophase is when they first become visible, and in anaphase, sister chromatids move apart. Now let's dive into some definitions. The centromere is the spindle fiber attachment site, while haploid refers to the chromosome number in gametes. Meiosis is the reduction division, and cytokinesis is all about dividing the cytoplasm. During interphase, cells prepare for mitosis, which occurs in roots and stems, while meiosis takes place in gonads. Chromosomes, the bearers of hereditary units, are visible during division, and chiasma is where crossing over happens. The centrosome is crucial as the division initiator in animals and organizes spindle fibers. Finally, karyokinesis is the division of the nucleus. And remember, anaphase is when chromatids finally move
DNA
apart.
replicates during S phase. True. During meiosis, the chromosome number is reduced to half. Also true. But hold on. Daughter cells of meiosis are not genetically identical. False. They show variations. Now, sister chromatids do separate during anaphase. True. But calling mitosis a reduction division, that's false. It's actually meiosis that gets that title. Prophase is not between divisions. False again. It's interphase that fills that role. And crossing over? That happens in meiosis, not mitosis. So that's false too. Keep these facts straight, and you'll ace your biology. DNA replication is a fascinating process that occurs during the S phase of the cell cycle. Imagine chromosomes aligning at the equatorial plate during metaphase, ready for their big moment. In meiosis, homologous chromosomes exchange segments, creating genetic diversity. This is crucial because variations can lead to the formation of new species. Did you know that the rungs of the DNA ladder are made of nitrogenous bases? It's like a secret code. Consider this analogy. Mitosis is to somatic cells as meiosis is to gametes. Just like ribosomes are essential for protein synthesis, centrosomes play a key role in spindle formation. And think about hydothodes and gutation, similar to how stomata are vital for transpiration. Finally, genes are to DNA what chromosomes are to the cell nucleus. Growth happens through mitosis, while variation thrives in meiosis. Isn't biology just incredible?
In the dance of cell division, we start at interphase, where the cell prepares for its big moment. Then, it's showtime. Prophase kicks off with chromosomes condensing, followed by metaphase, where they line up like soldiers. Anaphase pulls them apart, and finally telophase wraps it all up, creating two new nuclei. Switch gears to the cell cycle. G1 phase is all about growth. S phase is when DNA is replicated. G2 phase ensures everything's ready. And then we dive into M phase for mitosis. Now zoom in on nucleotides. They're made of deoxyribose and a base, forming nucleosides, which combine to create nucleotides, the building blocks of DNA. Each step is crucial, weaving the intricate tapestry of life. In the world of genetics, let's play a quick game of odd one out. First up, we have centromere, chromatid, centrosome, and chromosome. The odd one here is centrosome. It's all about organizing microtubules, while the others are key players in cell division. Next, we have genes, chromosomes, ribosomes, and nucleus. Ribosomes take the spotlight as the odd one out, since they're all about protein synthesis, while the others are all about genetic material. Finally, let's look at adenine, cytosine, penicillin, and thymine. Penicillin is the odd one out. It's an antibiotic, while the others are essential nucleobases in DNA. Keep your mind sharp and always look for the odd ones.
Chromosomes are like the ultimate data storage units, carrying our genetic information, the blueprint of life. Then we have the centrosome, the unsung hero that organizes microtubules, ensuring everything is in place for cell division. The centromere plays a critical role too, acting like a traffic cop to guarantee that chromosomes are accurately segregated during division. And let's not forget the spindle fibers, the dynamic ropes that pull apart chromatids, making sure each new cell gets the right genetic material. Together, these components orchestrate the dance of life at a microscopic level. Daughter cells in meiosis are unique, and that's thanks to crossing over. This process mixes up genetic material, ensuring no two gametes are alike. Meiosis is a reduction division, which means it halves the chromosome number, creating haploid cells. This is crucial because when fertilization occurs, those haploid gametes combine to restore the diploid number, giving rise to a new organism. The variations we see in sexually reproducing organisms, they stem from this genetic recombination during meiosis, driving evolution and diversity. It's nature's way of keeping things interesting. In the fascinating world of cell division, we encounter mitosis and meiosis, each with distinct stages. During metaphase, chromosomes align at the cell's center, while in anaphase they pull apart, ensuring each new cell gets the right genetic material. Prophase kicks things off, where chromatin condenses into visible chromosomes. Now, when we look at plant versus animal cells, the presence of centrioles is a telltale sign of an animal cell, and let's not forget crossing over during meiosis. This crucial process occurs at the chiasma, allowing segments of DNA to exchange, leading to genetic variation. This recombination is vital for evolution and diversity in life forms. The cell cycle is a fascinating journey where cells grow, replicate their DNA, and divide. It's like a well-choreographed dance. First, we have interphase, where the cell prepares for division, making copies of its DNA. Then comes mitosis, where those copies are neatly separated into two new cells. Don't forget about cytokinesis, the final split. Understanding this process is crucial, as it's fundamental to life itself. Mastering these concepts not only boosts your biology knowledge, but also helps you ace those exams. Keep revising, and you'll be ready to tackle any question that comes your way.